clinical issues. There's been a slight switch in the chair. So I'm actually going to be joined by Dr. Nanya Van Giel, who's going to co-chair this session with me. But to kick things off, we are going to have a great presentation. I know it's going to be great even before I hear it because it's coming from Dr. Amit Pandya. And, you know, he's going to give the presentation that, you know, all patients want to know about. You know, what's the latest in terms of therapy and what can we expect? So I'm not going to talk anymore because we want to hear from Dr. Pandya.
Thank you. Thank you very much, Harvey. It's a pleasure for me to be here to speak to you. All right. So can you see my screen okay?
Yeah.
Okay, great. Well, thank you very much, everybody. It's a pleasure for me to speak to you here, and greetings from California. It is 5:30 in the morning for Neda, Victor, and myself, and they look much more put together and awake than I am, but I'm going to do my best to talk to you today about the advances in the treatment of vitiligo with JAK inhibitors. And I want to thank the organizers of the meeting for inviting me to give this lecture, and thank you, Harvey, for that introduction. These are my disclosures. There are quite a few companies that are developing JAK inhibitors right now, and I am involved in consultation and activities with them. Well, you heard Doctors Harris and Seneschal yesterday talk about the pathogenesis of vitiligo and how does autoimmunity work in vitiligo. As you can see here, we know that interferon gamma produced by the innate and the adaptive immune system binds with the interferon gamma receptor on keratinocytes and Langerhans cells. And then what happens through the JAK-STAT pathway is the production of certain chemokines, CXCL10 in particular. These chemokines are the signal that the that the skin uses to attract the pathogenic T cells to the skin. These CD8 positive T cells that you've seen pictures of in several of the presentations that you've seen already in this meeting. Those CD8 positive T cells crawl up into the skin, they migrate to the area, to the basal area, they find the melanocytes, they secrete granzyme and perforin, and they kill them. And so that's the cause of vitiligo, this autoimmunity that we see. Well, if we can block that JAK-STAT pathway, then we can have some, perhaps some utility in eliminating these cells and improving vitiligo, and that's the logic behind JAK inhibitors. When it comes to vitiligo, I like to look at 3 pillars of therapy. One pillar is to reduce triggers. We know that when a patient falls down and cuts their skin, there can be a vitiligo lesion that occurs there. And so we use antioxidants to try to reduce the amount of reactive oxygen species that lead to the unfolded protein response and other activation. You heard John Harris speak yesterday about heat shock protein and other triggers of vitiligo. So we want to reduce triggers. We reduce triggers with antioxidants. We also want to avoid trauma to the area. The other thing we want to do is reduce the immune attack to the skin. The T that I just told you about, the CD8 positive T cells, we want to eliminate those from the skin. And that's one of the main focuses of our treatment as we see patients every day. We use corticosteroids, topical as well as oral. We use tacrolimus, pimecrolimus, and now we can use JAK inhibitors to reduce this immune attack of the skin. But notice that the JAK inhibitors are for this one column. That's their role, is to reduce that immune attack on the skin. Finally, we want to increase melanocytes and melanin production in the skin to try to repigment the skin. And of course, the mainstay that we use for that is phototherapy, either in the office or even sunlight, some type of UV exposure. There have been studies with afamelanotide, which stimulates the melanocytes to proliferate. It's an MSH analog. And then we use skin grafting as well as cellular grafting. And of course, this meeting is right now headquartered in India, which is one of the leading countries in the world when it comes to cellular and skin grafting. So these are the three pillars that we use to try to improve vitiligo. It turns out that phototherapy at the low doses will stimulate the melanocytes, but when you start going to higher doses, phototherapy itself can reduce the immune attack on the skin. When you use these three pillars for treatment of the patient, then you have effective treatment of vitiligo. And there have been questions from people in the audience who have asked, what if I only do that central pillar? Are we going to have success in the patient? And I believe that you have to have combination treatment in order to really have successful treatment of the patient. So that gets us to the JAK inhibitors. What are the JAK inhibitors? Well, these are small molecules developed to treat a variety of immune-mediated disease. They've been developed now for the last 10 plus years. JAK inhibitors or janus kinase inhibitors inhibit the janus kinase enzyme. Janus kinase enzymes are tyrosine kinases that associate with cytokine receptors on cell surfaces. So here you see in orange cytokine receptors, okay? And so for example, let's pretend that one that's in orange is a receptor for IL-23 and IL-12. Well, these enzymes associate with the cytokine receptors, and they, when the antigen binds to that, the JAK actually ends up getting cross-phosphorylated, and it activates the signal transducer and activation of transcription protein. That is what we call STAT. These STAT proteins then go from the outer surface of the cell to the nucleus and affect transcription. And then, of course, that causes a variety of effects that lead to pathogenesis of the disease. So there are four members of the JAK family: JAK1, JAK2, JAK3, and TYK2. Here you have where you see the heterodimers of JAK kinases that are associated with various receptors of cytokines. So let's look at the first one. What if I told you that IL-2 is associated with rheumatoid arthritis? Well, then maybe you would want to develop an inhibitor of JAK1 and 3 for rheumatoid arthritis. Well, that's exactly what Tofasitnib does. It's a JAK1/3 inhibitor. It is actually approved for rheumatoid arthritis and some other conditions as well. What if I told you IL-6 was important for systemic lupus? Well, then you would want to develop a product that has JAK1 inhibition, TYK2, JAK2 inhibition, and that's exactly what's happened. These are being used, these particular inhibitors, for the treatment of systemic lupus. Now, interferon is, we know it's important for vitiligo. You've heard a couple presentations yesterday from John Harris and Julian Seneschal talking about type 1 interferon, interferon alpha, beta, as well as type 2 interferon, interferon gamma. Well, these interferons are inhibited by JAK1 inhibitors, TYK2 inhibitors, JAK2 inhibitors. Now, erythropoietin and other cytokines that stimulate granulocytes and platelets, those are inhibited by JAK2 inhibitors, and so that's something that is going to affect the bone marrow, right? And so if you have a JAK2 inhibitor, you're going to have effect on white blood cell count, platelets, hemoglobin, and that's exactly what ruxolitinib oral does. It is the treatment for myelodysplastic syndrome and myelofibrosis because it has that specific effect in the bone marrow, but we may not want to have that effect in vitiligo. And then finally, IL-12 and IL-23, as you know, that is associated with psoriasis. And so JAK inhibitors that have JAK2, TYK2 inhibitors have been developed for psoriasis. So by understanding which of these heterodimers affect which receptors, you can start now imagining and thinking about which diseases you might want to use these various JAK inhibitors for. There are quite a few JAK inhibitors that are being developed for vitiligo. It's a very exciting time. I've been a dermatologist now for 30 years, and I have never seen a more exciting time in my life for the treatment of vitiligo. Oral Tofasitnib and topical Tofacitinib, JAK1/3, inhibition topical ruxolitinib cream, which is JAK1/2, topical ATI 5002, which is a JAK1/3, topical Certulatinib Gel, which is a JAK1/3-SIK, which is a different type of tyrosine kinase inhibitor, oral PF, the 1600 molecule, and oral PF, the 0841 molecule. The first one is a JAK3 inhibitor, and the last one is a JAK1 TYK2 inhibitor. All of these either have been studied or are in the process of being studied for vitiligo. Well, I'm sure that you all remember this article. This was now 5 years ago. It's hard to believe this has been 5 years now since this article was published. And this is an article by Brett King, in which they used Tofasitnib in one patient who had progressive vitiligo for 1 year. The tacrolimus and triamcinolone were ineffective, and the patient had 10% BSA affected. The patient was given 5 milligrams of Tofasitnib every other day, and after 3 weeks, it was increased to once per day. Repigmentation started at 5 months, nearly completed, at 2 months, nearly completed at 5 months, and 5% BSA remained depigmented at the end of the study. What that means is she got half of her color back, but you can see her forehead and the hands did very well, and there were no side effects in this patient. Tofasitnib tends to be pretty well tolerated. There is the potential risk of having increased infection, but in the rheumatoid arthritis literature, it's been pretty well tolerated. Now, moving forward 3 years, Brett King published this article of a 35-year-old Latin American female who had spectacular results. She had had vitiligo for 12 years, and she got Tofasitnib 5 milligrams twice a day, along with phototherapy. You can see her results are outstanding. And I think that the phototherapy definitely helped. She had over 75% repigmentation after 3 months, as opposed to the last patient who had 50% repigmentation. Now, this is quite an expensive medication. In the United States, it's around $4,200 per month for this medication. Brett King and John Harris and others, they published this article, which was a retrospective review of 10 patients in which these patients were treated with Tofasitnib 5 to 10 milligrams twice a day for 10 months, for a mean of 10 months. All 5 achieved repigmentation of sunlight-exposed skin or with low-dose narrowband UVB therapy. And here you see an example of that. Notice in this patient where the sun was hitting her, in this area, she got repigmentation. But where the sun was not hitting her, she did not get repigmentation. Now, John did sampling using blisters. So he creates 1 centimeter blisters on lesional skin, non-lesional skin, and he determines the levels of CXCL in the blister fluid. And he was able to show that CXCL levels decreased in sun-exposed and sun-protected lesions. Remember, those sun-protected lesions did not improve. So you reduce the CXCL, but the lesion did not improve. This is one more bit of evidence showing that you need to have combination therapy in order to really see a great improvement in these patients. Combination with some type of UV exposure. Well, Tofasitnib can also be used topically. Here you can see that 2% Tofasitnib was used in 11 patients from our center. And these patients were unresponsive to narrowband UVB and topical steroids or topical tacrolimus. Now, these are usually low potency topical steroids or mid potency topical steroids. They were treated with Tofasitnib cream for 3 months plus narrowband UVB or sunlight to the face. And you can see the VASI improved by 70%. Baseline VASI was 0.8. Now, the size of the patient's hand is 1%. So 0.8 is a lot of the patient's face. So it went from 0.8 to 0.23. I would like you to notice on this man's face that he has hundreds of confetti-like lesions. He also has that hypopigmentation known as hypochromic or trichrome vitiligo, like we like to call it in the United States. He has signs of activity. Those are your clues. Those are your clues that this patient has an infiltration of CD8 positive T cells in the skin, and that he would be likely to respond to products that are going to eliminate those cells from the skin, unlike long-standing segmental vitiligo, which usually does not have those cells in the skin. Another example of a patient who had been resistant to narrowband plus steroids or tacrolimus who did respond to the Tofasitnib cream. And here you see the other side of the face with good results. And she has the trichrome vitiligo at baseline. Another example of a patient who did well using this combination of narrowband UVB and Tofasitnib. You can see how much improvement there was with this combination. The skin below the nose, the skin around the mouth, the lips, the eyelids, the corners of the mouth tend to be resistant areas, but you can see in her non-resistant areas, she had good results. This is an interesting patient who has mixed vitiligo. Mixed vitiligo is rare, but I certainly have seen these patients in my practice. And mixed vitiligo means that the patient has segmental vitiligo, but then developed non-segmental vitiligo. And you can see her non-segmental vitiligo did fantastic, but even her segmental vitiligo improved a little bit, as you can see here. So another case of Tofasitnib and narrowband UVB. So these are simply case reports, case series. We need larger studies, control studies with topical Tofasitnib to see if it's going to be effective for vitiligo. So that gets me to topical ruxolitinib, and you've already heard a couple presentations about topical ruxolitinib. This started when David Rosmarin treated 9 patients and published this article. He used 1.5% ruxolitinib cream twice a day for 20 weeks in these patients. The 4 patients that you see here in the picture who had facial involvement had a 76% improvement, fantastic improvement. So these patients were getting, we suppose, light exposure. David lives in Boston, so they were getting light exposure in Boston, and that was supposedly helping them. Notice that the overall VASI score, which includes the chest, the back, proximal extremities, there was only a 23% improvement in the overall VASI score. And so it was thought that maybe UV exposure is likely necessary for success. These patients did not have side effects. Now, the phase 2 study has been completed in the United States, and you've already seen a couple presentations of the phase 2 study in which 157 patients with over 0.5% facial involvement, 3% non-facial involvement were treated, and we looked at outcome measures such as the F-VASI. Did you know that the F-VASI was actually developed for this study? This was the first study where they really developed the F-VASI, and the F-VASI was developed because we saw in David Rosmarin's study that the face was preferentially improved. And so we wanted to know, well, is there a measurement, outcome measure that we can use just for the face? And so the F-VASI is one that has been used, and that's what was used to look at the 24-week and the 52-week results. We also looked at the Physician Global Vitiligo Assessment of clear or almost clear at week 24. You've seen this slide before, but I want to point out a few things. I would just want to let you know, based on this slide, I think this is a group of patients that were difficult to treat. And here's the reason I say that. The average age of vitiligo patients is 30. These were older patients, and as you know, older patients are harder to treat. Patients around the world are normally, the majority of the world is brown skin, right? And yet you can see that the majority of these patients were skin types 1 through 3, and they tend to be harder to treat compared to darker skin patients. Look at the baseline F-VASI, 1.26. That means over the size of their hand involvement of the face. That's a lot of involvement of the face. Also, look at their total VASI was 18. You heard Dr. Van Giel talk about what is considered mild, moderate, and severe vitiligo yesterday. Well, 18 is pretty bad. You heard her talk about 1% and 5%. This is 18%. These are hard patients to treat. Finally, look at the disease duration was 14 years. These are not patients who had just had vitiligo the last year. These are patients with a lot of long-standing disease. And then you've seen this result, the F-VASI 50, which means that 50% of the color came back on their face, you can expect that after 1 year. You can expect that almost after half a year. You can see 45% got F-VASI 50, but F-VASI 75 was 50% after 1 year. And if you know about the info study we did, patients really want 75 to 80% or more repigmentation to be considered satisfactory. This is our goal, F-VASI 75 to F-VASI 90. And you can see after 1 year, we've achieved that in half the patients on the face. Now, we have less of a response when it comes to the total body, but when you look at the patients who were only treated in 20% of their body, then you can see that we had better results on the total body. Why do we look at 20%? Because this drug it wasn't allowed for the patients to apply it to more than 20%. Remember what I told you about JAK2 inhibitors do affect the bone marrow. This is a JAK2 inhibitor. We want to be careful. We want to be safe in these patients. So we're applying it to less than 20% of the body surface area in these patients. If we do that after 1 year, we almost get half the patients get a T-VASI 50. Now we want, of course, a T-VASI 75, 80 or higher. And that's going to be one of the goals, perhaps with phototherapy. Here's some examples of images of patients who were treated after 24 weeks. And here you can see another patient. She was on placebo. She was my patient. And you can see for 6 months, this patient did not improve. Very difficult for her to continue in the study, but she did it. And I commend her for it. In fact, she got worse during the first 6 months because she was on placebo. But look what happened to her after the second 6 months. The second 6 months, she transformed. And this was just once a day dosing. Now, I believe twice a day dosing is better, but in her, even once a day dosing caused a 93% improvement in her facial VASI, something that was quite spectacular. And she and her husband were delighted with this result. Here's an elbow that showed improvement with follicular repigmentation. So in terms of side effects, the main side effect I want to point out is that there were no really any serious side effects that were of concern and compared to overall in the study. But it was mainly acne. There was an increase in acne in patients, but they continued on the study and the acne got better. All right, so to summarize, I think there was substantial facial and total body repigmentation after week 24, continued improvement through week 52 with the highest dose. There was longer duration of therapy. There was near complete facial repigmentation, substantial body repigmentation, and all doses were well tolerated. The phase 3 study is ongoing. There was a question as to when this is going to come out. And so when the phase 3 results are reported next year, hopefully then that will proceed to FDA approval. All right, well, what other JAK inhibitors are there? Well, there's a JAK1/3 inhibitor called Topical Ati 5002. And in 2017, it was announced that there was going to be a pilot study of this phase 2 study in a solution applied twice daily in patients with non-segmental vitiligo. And 23 patients were treated for 24 weeks in this study. And if you look at the clinicaltrials.gov website, you will see that they reported that there was no improvement in the VASI score or the vitiligo noticeability scale in this study. And so this was a negative study with this JAK1/3 inhibitor. What about Certulatinib Gel? Certulatinib Gel is a JAK1/3 and a SYK inhibitor, which is another tyrosine kinase. And it was announced in December 2019 that this particular study was going to be enrolled. It was a phase 2A study, and this gel was going to be applied twice daily to vitiligo lesions in patients with non-segmental vitiligo. And 33 patients were treated for only 7 weeks. This was a tolerability and biomarker study. It was mainly used to look at the skin to determine what was going to happen to the biomarkers in the skin and tolerability with this gel. And at this time, there are no results posted yet. So we'll be looking forward to the results of that study. There are two oral studies being done, the PF 1600 and the PF 0841 for patients with non-segmental vitiligo. The first one is a JAK3 inhibitor, and the second one is a TYK2 JAK1 inhibitor. And if you remember in my slide, TYK2 JAK1 looks like it would be of utility for vitiligo. There's a phase 2b study in patients with non-segmental vitiligo that is being done. 366 patients treated for 24 weeks with a 24-week extension, and the enrollment is completed. And if you go to clinicaltrials.gov, you will see this information that I've just posted here on the left. And so far, the results haven't been posted. But since enrollment has been completed, and since it's a 24-week study, then you can anticipate the results of this trial coming out in the not too distant future. This is exciting because that's what patients want. Patients want something that's convenient and easy and safe to take for vitiligo to treat their whole body. Because it's a TYK2 JAK1 inhibitor, then we're not expecting bone marrow toxicity. We're not expecting much toxicity. So it'll be exciting to see the results of this study. So I just want to summarize that so far, there are several JAK inhibitors that are being studied and that you can find online at clinicaltrials.gov. There are others that I haven't discussed because the information is not public yet, that are also being studied for vitiligo. These are being studied for many other diseases, and of course, when they are used for atopic dermatitis, ulcerative colitis, psoriasis, then people are going to start thinking about vitiligo because there's such an unmet need. We know that these inhibitors inhibit the pathogenic T cells that target melanocytes. They show promise in the treatment of vitiligo, but I believe it's optimized when you combine it with UV light exposure. Multiple studies are underway to determine the optimal JAK-TYK2 inhibitor, the method of delivery, oral versus topical, solution versus gel versus cream versus ointment, the dosing in terms of concentration, the dose of oral medications, how many times a day should it be used, and then also its use in combination therapy, particularly with phototherapy. And I will stop right there and thank you for your attention. I'm happy to take any questions. I would like to acknowledge several people that have helped me over the years. Certainly John Harris and his lab and my collaboration with him has helped me as a non-basic scientist understand the science of vitiligo. In the United States, many people, including the ones you see here, that have helped me. So many people internationally, and I miss all of you during the COVID pandemic. I wish I could see all of you because we as a family can work really together. Our fellows, our students, and especially our patients. Thank you very much for your attention. I'll stop right there.
Okay, great. Thanks a lot, Amit. I totally agree with you that, 5 years ago, this talk could have been finished in 5 minutes, but now in 2020, we could do the whole morning or the whole day on JAK inhibitors. But there's a bunch of questions, but one question I know that is on patients' minds and on dermatologists' minds are, our patients are impatient for getting access to this stuff, and our derms, they like to compound and they like to play around with new molecules. So what's the current kind of status of off-label use of topical, compounded creams? It's not official, but I'm sure it's happening out there.
Right. Well, I'll tell you, thousands of patients in the United States are being treated with compounded creams by thousands of dermatologists. This information has really caught on. As I speak to general dermatologists, just about everybody knows about JAK inhibitors for vitiligo. There are multiple pharmacies that are compounding this. Now, some of them may be compounding it from the pills that are available that for rheumatoid arthritis. Some of them may be compounding it from powders that are available from other sources. Whatever the case is, they seem to be doing a good job in making a smooth, non-granular cream for the patients to apply. The cost varies depending on one pharmacy to another. Insurance companies do not tend to cover this. However, patients are willing to do this, number one, because of the enthusiasm of their physicians, and number two is because we're starting to see the results.
Okay, so it's definitely out there and people are using it like crazy. I had
I
a,
think we, yes, sorry, I think
so I think
more
we need,
questions. Yes.
yeah, so maybe we have time maybe for just one or two more
One,
questions.
okay. I think one question can be interesting to answer. I think this is returning over and over again. So the comparison with tacrolimus, we know that the evidence or the studies are missing yet, but in your experience, do you have an impression? This was also a question we received actually in the chat box. So what is your personal experience if you compare those two in the topical form?
Well, my personal experience with tacrolimus has not been as good as what has been reported. About 10 years ago, we treated 2 target lesions below the neck, one side with tacrolimus and one side with Vaseline. And we were blinded and we did phototherapy and the results were equal in both. And so I think for tacrolimus, the face is probably more effective. But as I'm sure you have had the same result, I have seen hundreds of patients who come in and say, "Doctor, I have been treated with a topical drug called tacrolimus and it did not work." And my own experience is that the topical JAK inhibitors work better than tacrolimus or pimecrolimus.
Okay, thank you. Do we still have time for more questions?
Unfortunately, I don't think we do because we need to kind of move on. So, but I mean, obviously, Dr. Pandya, you have inspired a lot of questions. Maybe there's a way to pick those up in written form somehow, but and maybe some of these questions will come up later during the symposium, but I guess we need to move on. So I will hand it over to you, Nanya.